Welcome everyone to our virtual lab. I'm really glad you could join me. Today, we're going to dive into a very important specimen, the nematode parasite, Ascaris. This is a classic in any zoology course, so it's one you really need to know well. Let's get started.
Now, everything we're going to cover today comes directly from your official practical course materials. That's the Z102 General Zoology course from Mini University. So think of this as a focused study session. We are going to hit all the key points you absolutely have to know for your lab work and, of course, for your exams.
Okay, so here's our game plan. We're going to follow the same steps you would in a real lab. First, we'll start with the big picture, how we classify this worm. Then, we'll zoom in to look at the outside to see the differences between males and females. After that, we'll do our virtual dissection to see the internal systems, take a look at a cross-section, and we'll finish up by reviewing the most important English keywords that you need to remember.
Before we do any cutting, even virtual cutting, a good scientist always starts with classification. It's so important to know exactly where our specimen fits in the grand scheme of the animal kingdom. So that's where we'll begin.
Right. Ascaris is in the kingdom Animalia, of course, and the subkingdom Metazoa. But the rank I really want you to focus on today, the most important one, is its phylum, Nematoda. This is the massive group of roundworms. I want you to burn that word into your memory, Nematoda. From there, it's in the order Ascaridata.
Okay, time for our first real observation. We're looking for something called sexual dimorphism. That's just a scientific way of saying the visible differences between the males and females of a species. This is always your first step in the lab when you get a new specimen. And here they are, the adult male and female right next to each other. The easiest way to tell them apart is to look at their posterior ends, their tails. See the female on the right? She's usually bigger, and her tail is straight. Now, look at the male on the left. See how he's smaller and his tail has that very distinct sharp curve? That curve is a dead giveaway, and it's all about reproduction.
So let's just nail down that key difference. The male's posterior end is curved because it holds these special structures called copulatory spicules, and that is a very, very important English keyword for you. Say it with me, copulatory spicules. They're used during mating. The female, on the other hand, is larger, she doesn't have these, and her posterior end is straight. Simple as that.
Excellent. So now we can tell a male from a female just by looking at them. It's time for the really interesting part, our virtual dissection. Let's open them up and take a tour of the internal anatomy.
We'll start with the male. First, let's trace the digestive system. You can see it's really just a simple, straight tube, basically a gut that runs from the mouth all the way to the tail. Food goes in the mouth, gets digested in the midgut, and waste leaves through the cloacal opening. For a parasite that lives in a sea of food, it doesn't really need anything more complicated, does it?
Now, the system that really dominates the inside of the male is the reproductive system. Look at this incredibly long, coiled-up tube. That's the testis. This leads to the vas deferens, which carries the sperm to a storage area called the vesicula seminalis. From there, it goes through the ejaculatory duct and finally out through those copulatory spicules we mentioned. Let's practice those English terms. Testes, vas deferens, vesicula seminalis.
And this brings us to another key feature of the male, the cloacal opening. This is an essential keyword. It's a single exit, a common opening for both the digestive waste and the reproductive material. You need to remember this because it's a huge difference from what we're about to see in the female.
All right, let's shift over to the dissected female. The digestive system, that gut running down the middle, looks pretty much the same as the male's, but wow, look at the reproductive system. It is absolutely massive. It's so much more complex, and it fills almost the entire body. And there's a very good reason for that. Her main job is to produce a truly staggering number of eggs.
So let's trace the path here in the female system. Right away, you should notice a huge difference. Many of her organs are paired. See, we have paired ovaries that produce the eggs. The eggs travel through the oviduct to the paired uterus. Now, after mating, sperm from the male is stored in the receptaculum seminis. Finally, all those fertilized eggs pass through the vagina and leave the body through a totally separate genital opening. This table is a fantastic summary of those key differences. So let's recap. The male has a single testis. The female has paired ovaries. Sperm is stored in the male's vesicula seminalis and in the female's receptacle seminus, and the big one. The male has that all-in-one cloacal opening, while the female has a separate genital opening just for reproduction.
Okay, great job so far. We've seen the worm from the outside, and we've looked at it cut open lengthwise.
Now we're going to take a completely different view. We're going to virtually slice it right across to make what we call a transverse section, or TS for short. This view is perfect for seeing how everything is arranged inside.
So why bother making this kind of cut? What new information can a cross-section really tell us? Well, it reveals the worm's internal architecture. It shows us the relationship between the body wall, the body cavity, and how all of those organs are packed so tightly together inside.
Let's start with the parts that both the male and female have in common. I want you to focus on the outermost layer of either of these diagrams. That thick, protective layer is the cuticle. Right underneath that, you have the epidermis and then a layer of longitudinal muscle. Also take a look at the dorsal, ventral, and lateral lines. These are important features you'll find in the body wall.
Now for the good part. Let's look inside the body cavity, which, by the way, is a pseudocoelom. This is where you see the biggest difference. In the male cross-section on the left, you'll see a few circles, which are sections of the single testes and maybe the large vesicula seminalis. But look at the female on the right. It is packed with circular structures. These are all the different sections of her paired ovaries, her oviducts, and those very large, thick-walled uteri, which would be full of eggs. It's much more crowded in there.
We have covered a lot of anatomy today, a lot of detail. And to do well in your zoology practicals and exams, you must master the English scientific keywords. It is absolutely essential. So let's do a quick review of the most important terms from today's session. Please take a moment, look at these words on the screen. Let's practice saying them. Sexual dimorphism, copulatory spicules, cloacal opening, uterus and testis, lateral lines, cuticle, midgut. Knowing what these words mean and how to spell them is crucial for your success.
And I'll leave you with one final question to think about. We saw a really simple digestive system, but we also saw these massive, incredibly complex reproductive systems. So my question is, how does this specific anatomy, this intense focus on making babies above all else, help make Ascaris such an incredibly successful and widespread parasite? That is the perfect thing to think about before your next lesson. Thank you very much for your attention today.